DNA contains all the information needed to make and maintain a living organism. Some DNA, known as coding DNA, is transcribed into messenger RNA, which leaves the nucleus of a cell and is later translated into amino acid chains that hold to become proteins. Only 2% of DNA is coding DNA. Though the other 98% of DNA used to be referred to as junk DNA, it actually serves vital functions for living organisms. For example, this DNA contains regulatory sequences that enable certain transcription factors to bind to DNA, allowing DNA to be transcribed. If such transcription factors, like RNA polymerase, are unable to bind to a strand of DNA, then that strand of DNA won't be transcribed into messenger RNA. There are different mechanisms that can affect the way DNA is transcribed without altering DNA sequence. The term epigenetics refers to any one of these mechanisms. Methylation is an example of an epigenetic mechanism. Methylation refers to the adding of a methyl group to a nucleotide base, usually cytosine, though sometimes guanine. The enzyme DNA methyltransferase catalyzes this reaction, in which a methyl group, CH3, is added to the 5-carbon of cytosine, forming 5-methylcytosine. The addition of this methyl group prevents transcription factors from binding to DNA, preventing this DNA from being transcribed into messenger RNA. Scientists used to think that methylation only occurred during field development, when cells are being differentiated into different tissue types. But we now know that methylation can occur during a person's adulthood. Methylation can change due to factors like diet or the environment. An experiment conducted by molecular biologist and geneticist Moji Snaith and neurobiologist Michael Maney has shown that parenting can affect the way one's DNA is methylated. In an experiment, Schniff and Maney took rats that were either highly attentive mothers or rats who were distant mothers, and then absorbed their offspring. Once the offspring were fully developed, they would examine the rat's hippocampus. The hippocampus is the part of the brain that's responsible for regulating the stress response. These researchers found that in the brains of rats with distant mothers, the genes for glucocorticoid receptors were highly methylated, while the same genes in rats with loving mothers lacked the same methylation. This means that the signaling pathway for glucocorticoid wasn't functioning the way that it should be in the brains of rats who had distant mothers, making it harder for these rats to cope with stress. So it's definitely interesting to think that something like social interaction can affect the way someone's genes are transcribed and expressed. It's definitely interesting to see how the field of epigenetics changes over the next couple of years.